Hello friends, in this video we'll be discussing about the chapter cell. So let's start with the introduction of cell and the basic question, what is a cell? Cell is the fundamental structural and functional unit of all living organisms. The fundamental structural as well as functional unit. Anything less than a cell cannot exist as has no independent existence. Like anything less than a complete structure of a cell does not ensure independent living, because it is the cell which has all the metabolic functions, all the functions of assimilating energy and doing all the reactions which are essential for life. So let's see what are the unicellular organisms capable of. They are capable of independent existence just because they can perform all the functions which are essential for life. So now let's see the scientists who have contributed to the research of cell biology. First of all, the scientist who came was Anton von Leeuwenhoek. He worked at a shop of lenses, the lenses which are used for your eyewear. So he was very curious to see minute details of cork which was there on the champagne bottle. So he took that cork and made his own microscope using some lenses, and he was the first person to see as well as describe a living cell. Then came Robert Brown. He was another scientist and he worked specially on nucleus. So he discovered the nucleus, which is the main controller of the cell. Then let's compare the two microscopes. First of all, the initial microscope had very less magnification power and could barely see a cell. But later on, with advancements came compound microscope. This microscope was that much big that it required a space of a room, and this produced that much magnification. Like earlier it was not possible to see a cell, but this compound microscope can enlarge a cell and show the two membranes of mitochondria present in that. So that was that much magnification it could produce, and this revealed all the structural details of the cell. So now the whole chapter which we will be studying right now is dependent on the studies of electron microscope done for the cell biology. Now let's study about the cell theory. Three scientists paid their life to develop a theory called cell theory. It was the first scientist who started his studies regarding the cells was Matthias Sheldon. Sheldon was a German botanist. Botanist is a person who studies plants. So he examined large number of plants, and he observed that all plants are composed of different kinds of cells. So he said plants had many cells. It is not like that a single type of cell is making whole plant body. And he also said that these different kind of cells make tissues of the plants. So he did his research in year 1838. It is mentioned in the NCERT, so you need to remember this. In 1838, Matthias Sheldon, who was a German botanist, did research on many plants, and he found that plants are made up of different types of cells, and these cells form tissues of the plant. Then in the consecutive year, like in 1839, came another scientist, Theodor Schwann. He was a zoologist, a person who studies about animals. So he studied about different kinds of animal cells, and he revealed the most important detail of a cell. He said that all cells are covered by a layer, outer layer, which is now known as plasma membrane. So which scientist told about who described plasma membrane for the first time? It was Theodor Schwann in year 1839, the year just after Matthias Sheldon. He also studied few plant cells, and he told that cell wall is a unique characteristic of plants. So it was not a botanist who discovered the cell wall of a plant cell. It was a zoologist named Theodor Schwann who discovered cell wall of plants. Schwann proposed the hypothesis that bodies of animals and plants are composed of cells and products of cells. Because he did research on both animals and plants, so he said that the bodies of both animals and plants are made up of cells and products of cells. Then at that time in 1839, a cell theory was proposed. That cell theory was incomplete in the sense it did not tell about how new cells were formed. They told that animals and plant bodies are made up of different kinds of cells and products of cells, but they had no idea how these cells are formed. Then came another scientist, the third scientist who contributed in cell theory, and his name was Rudolf Virchow. He in year 1855 told that no need to panic, bro, because the cells are formed from the pre-existing cells. One cell multiply into the daughter cells, and consecutively the daughter cell will multiply into its daughter cell, and this way new cells are formed from the pre-existing cells. So he first explained that cells divided and new cells are formed from pre-existing cells. He gave a sentence for this whole theory, which was called as omnis cellula e cellula. This sentence means that new cells are formed by dividing from the pre-existing cells. And he modified the hypothesis of Sheldon and Schwann and gave a final cell theory, which included the points of Sheldon and Schwann, which was like all living organisms, be it animal or plants, are composed of cells and products of cells. Cells and product of cells. And the second point which he added was that cells arise from pre-existing cells. So just recapping, the Sheldon, which was a botanist, he told that plants have different kinds of cells, and these cells form tissues of plants. Then came Schwann in 1839. He told that animal cells have an outer limiting membrane, which is called as plasma membrane, and presence of cell wall is unique characteristic of plant cells. Then 1855 came Rudolf Virchow. He said that cells arise from the pre-existing cells and gave a sentence which was omnis cellula e cellula. And this way the cell theory was formulated by the work contribution of three scientists. Now let's see an overview of cell. Plant cell has a distinct cell wall as its outer boundary, and within cell wall it has cell membrane. But the cell wall is unique for plant cell and is absent in animal cells. So therefore animal cells have outer membrane as the cell membrane as an outer delimiting structure. Delimiting means making a boundary of animal cell. And inside each cell we have dense membrane bound structure, the structure which controls the functioning of cell, which is known as nucleus. Nucleus contains chromosomes, and we know that human cell contains 23 pairs of chromosomes, and these chromosomes contain genetic material, and this genetic material is the DNA, which is deoxyribonucleic acid. But friends, prokaryotic cells do not have a membrane bound nucleus. So where is the genetic information of prokaryotes stored? It is stored in the form of nucleoid. Nucleoid is not a membrane bound structure which is present in the cytoplasm of prokaryotic cells, which stores the genetic material of prokaryotes. And whensoever somebody tell you about prokaryotic cells, just remember three groups. First is bacteria. Then comes cyanobacteria. Cyanobacteria is blue-green algae, and the group is mycoplasma, PPLO. PPLO stands for pleuropneumonia like organisms. So these are the smallest known organisms present on earth, and they lack cell wall. We'll study them in detail. Now for the time being, let's study cytoplasm. Cytoplasm is present in both prokaryotic and eukaryotic cell. It is semi-solid in matrix. It is not liquid, it is semi-solid. It has firm consistency. It occupies the volume of cell and is the main arena. Arena means a space where metabolic reactions take place. It is present in both plants and animal cells, we have already discussed. So for cytoplasm, just remember that it is present in either be it a prokaryotic cell or a eukaryotic cell, cytoplasm is present. It is semi-solid in consistency, not liquid. And it provides space or say arena for cellular activities or metabolism reactions. Now let's discuss organelles. What do you mean by organelles? Organelles are membrane bound structures present inside the cytoplasm, which have their individual roles for the maintenance of life of a cell. So besides nucleus, besides means in addition to nucleus, eukaryotic cells also have other membrane bound structures which are known as organelles. And let's name few organelles present in eukaryotic cells. Remember that prokaryotic cells lack, that means do not have such membrane bound organelles. So these organelles are present, are distinctive characteristic feature of eukaryotic cells. So let's name few organelles. First is endoplasmic reticulum. It is the highway of cell because it provides an elaborate network within the cytoplasm to transfer the packed goods to different parts of the cell. Then comes Golgi complex. Golgi complex is the post office of the cell. It packs the thing which is to be transferred to different places inside the cell. Then comes lysosomes. Lysosomes contains enzymes for digestion, therefore they are digestive bags of cell. Then comes the mitochondria. It is a powerhouse of cell and provides ATP, that is the energy currency, adenosine triphosphate, used by every living organism. Then comes the microbodies and vacuoles. Vacuoles are like almirahs of cell in which cell stores its food and other important metabolites. Now let's discuss about ribosomes. Ribosomes, as we know, are the protein factory of the cell. They are non-membrane bound. We'll have to remember the organelles which are non-membrane bound, and first one is ribosomes. The second one is centriole, which we'll discuss just after ribosome. So ribosome is non-membrane bound, and its characteristic feature is that it is present in both prokaryotic as well as eukaryotic cell. It is present in both prokaryotic and eukaryotic cell. Ribosome in eukaryotes is present in cytoplasm as well as in organelles. So it is present in cytoplasm and also in organelles, within two organelles and upon one organelle. Within it is present in chloroplast. Chloroplast is the structure which provides green color to the plants, which contains chlorophyll. And another thing is mitochondria. So mitochondria is the powerhouse of the cell, and ribosomes are present within these two organelles, chloroplast and mitochondria, and it is present on the structure of rough endoplasmic reticulum. Friends, the endoplasmic reticulum, which is the highway of cell, if I draw endoplasmic reticulum, if ribosomes are present on its structure, then we call it rough endoplasmic reticulum, RER. But if ribosomes are absent on its structure, we call it SER, smooth endoplasmic reticulum. So if somebody asks, ribosomes is present inside how many organelles? Inside it is present of two organelles, which is chloroplast, the solar panels of cell, which trap solar energy and produce food, and the second one is mitochondria, the powerhouse of cell. And ribosomes are present on the structure of rough endoplasmic reticulum. Understood? So this is the line which I have written here. Ribosomes are present within two organelles, chloroplast and mitochondria, and on one organelle, which is rough endoplasmic reticulum. Now let's discuss the second non-membrane bound organelle, which is centriole. It is present only in animal cells, but to be very precise, it is present in some lower plants also. But if this is the NCERT statement, and if it comes in true false, this is a true statement that it is present only in animal cells. It is another non-membrane bound organelle, and it helps in cell division. So three things you need to remember about centriole is it is non-membrane bound organelle, just like ribosome. Second thing, it is present only in animal cells, and third thing is it helps in cell division. Now let's learn different sizes of different types of cells. You need to pay attention that in NCERT text the size is given different, and in NCERT the size is given different. So you need to remember both sizes individually. If the question is from the diagram, you need to write the values which were mentioned in NCERT diagram, and if the question is from text, you need to write the values from the text. So mycoplasma, which are the smallest cells, also known as PPLO, has a size of 0.3 micrometer as per the text. But in the diagram, PPLO is mentioned to have a size of only 0.1 micrometer. So in text it is 0.3 and in diagram it is 0.1 micrometer. Now let's see another example, which is of bacteria. Bacteria in text is mentioned to have a size of 3 to 5 micrometer, but in the diagram it is given only 1 to 2 micrometer. So 1 to 2 micrometer in diagram and 3 to 5 micrometer as per NCERT text. Now let's see third example, which is an egg of an ostrich. Ostrich is the flightless bird, the largest bird which is present on earth, and the isolated single, it is the egg of ostrich. Egg of ostrich is the largest isolated single cell. Largest cell is the egg of ostrich. Then comes the most studied cell, which is human RBC, red blood cell, which carries oxygen in hemoglobin. The size is 7 micrometer, and then comes the longest cells, which are nerve cells, also known as neurons. They are longest cell. Now let's see the diagram. Viruses, which were not mentioned in the text, have a size of 0.02 to 0.2 micrometer. Some people may wonder that PPLO are called as smallest living organisms, but viruses have a size smaller than PPLO. So why they are not told as the smallest living cells? Because viruses are not truly living. Not truly living. They are living only once they infect a host cell and take over the mechanism of their replication and metabolism, not truly living. And now if we compare the size of a bacteria to a typical eukaryotic cell, we can see the size is almost 10 times. Eukaryotic cell is 10 times larger than a typical bacteria. A bacteria has a size of 1 to 2 micrometer, whereas a eukaryotic cell has a size of 10 to 20 micrometer. Now let's discuss about the different shapes of cell. There are 6 different shapes of cell. The first one is disc-like. Then comes the polygonal. Then we have columnar. Fourth one is cuboid. Fifth one is thread-like. And sixth one is irregular. So we have 6 shapes. First is disc-like. Second is polygonal, then we have columnar, third one is cuboidal, fourth one is cuboidal, fifth one is thread-like, and sixth one is irregular shape. So these are the 6 shapes of cell, and the important line, which is shape of a cell vary with the function they perform. Each cell take up a different shape as per the function they are performing. For example, let's see individually. Human RBC are round and biconcave. Biconcave shape is this. This shape provides large surface area for the exchange of gases to the RBC, and RBC perform the function of exchange of gases. So therefore they have taken a shape in which their surface area can be increased, and which is biconcave shape. Then comes the WBC, white blood cells. They are the warriors of cellular life. That means whensoever there is an infection inside our body, WBC fights with the infecting organism, the pathogen, and prevents infection, further infections. They are amoeboid in shape, that means irregular shaped, because they need to pass through many capillaries and be present at the site of infection. Therefore their shape is amoeboid. Then comes columnar epithelial cells. Epithelial cells do not have any space between them because they form a compact lining of any tube inside our body, any tube or organ. So these are columnar epithelial cells. Now let's see about the nerve cell. Nerve cell is branched and long. They are long just because they need to transmit impulses to different parts of the body, and also they are branched so that they could interact with other neurons and form a elaborate network. And now let's see about tracheids. Tracheids are present in plants, and in plants it is present in xylem. Xylem is a tissue which is involved in transportation of water. So therefore they are elongated so as to transport water to far off places from roots to different parts of the plants. Then comes the mesophyll cells. Mesophyll cells are round and oval. Actually, if we see, mesophyll cells are a part of leaf inside plants. And mesophyll cells are of two types, which we'll have to study in the anatomy of plants as per NCERT. One is spongy cells and the other one is palisade cells. These cells which are shown in the diagram are spongy cells because they are round in shape. Palisade cells are elongated cells. So with this we have finished the introduction of cell biology. Hope you like this video. Thank you for watching.